Well, thanks to Manita and Applied Biosystems, as they're called now, for inviting me to this meeting and for putting on a really fantastic meeting so far. Some very good presentations and a really good venue. I have to be careful with this one. It's a bit more stable, is it? Okay, thank you. So I'm going to be reporting some collaborations we've done with Waters Group in Innsbruck and the University of Cologne, Peter Schneider's lab. Some collaborations we've done with Runa, Dennis and Roland in Melbourne. And we're going to finish with a few slides looking at a collaboration within Neo4gen, which is slightly more labs. So I haven't put up many slides looking at the Genomics Revolution because that's been well covered, particularly by Bruce this morning, first thing. But this is an interesting poster which you can still obtain from Nature, which really highlights the reduction in the cost per base. But you could reverse this trend line in the other direction, going up in terms of the amount of sequence that's generated from the technologies. So as we've moved along from the first human genome towards where we are now, we're getting more and more sequence for smaller and smaller areas. As Manfred said, there's this shift towards massively parallel sequencing. And we really sort of hooked into this in 2001 in SNP for ID and used this data, this genomic data, to start to choose SNPs. I borrowed a graph from one of Bruce's presentations, just to concentrate on the right-hand side of the graph. And we've really started to see a big reduction in cost, and this, as I say, this parallels the sort of increased coverage. And this has been brought about by the 454, which is no longer available, but also principally by Illumina HiSeq and LT Solid, and these have been extended into the systems that we've been talking about today and tomorrow, MiSeq and Ion PGM. Okay, so that's the sort of background in a nutshell. We're looking at SNPs using massively parallel sequencing. What are the considerations that we need to bear in mind when we start to sort of build a validation program? Well, I come from a background that uses snapshot for typing SNPs in a forensic context, but other techniques for typing SNPs in medical applications. And there are always good SNPs and bad SNPs. So one of the first questions we could ask ourselves is, if we're using this new technology to use SNP typing, are there good SNPs and bad SNPs, or not so good SNPs? And does this affect their performance if they're bad SNPs, or they don't produce sort of the best quality results? Does this affect their performance in low-level DNA? And we're interested in analysing degraded DNA in a forensic context, so we want very short amplicons. If some are not so good and don't work so well, we can swap them out. There's really no problem in an identification panel in taking some SNPs out and replacing them with better performers. But if we're interested in predicting externally visible characteristics and some of the better ancestry markers, they need to work for the information to be good quality. So are all SNPs in the multiplex equally well genotyped with Ion PGM or any other massively parallel sequencing technology? What is the genotyping precision? But also, as a corollary to that, what is the true genotype? So if we're measuring the precision with which a genotype is called, what's the real genotype? Nah. Let's leave that. And what happens to the sequence data when we put in low-level or mixed DNA into the system? There's another layer of complexity because we also have to talk about alignments. We have to consider the process of alignment. And we're comparing the generative sequences to a reference sequence. And sometimes there are problems with the context sequence to make a reliable alignment. And I've put an example here of a SNP that works perfectly okay with snapshot. It's one of the 52-plex SNPs, because we dodged the homopolymeric section on the right-hand side of the SNP base in the extension primer, so this is not an issue. But for an alignment, something like this causes a lot of problems. And this is something that Bruce Badoli's group and Niels Morling's group have both identified as a bad SNP. The other thing that's sort of difficult at this stage is we have to sort of get the balance right between throughput and quality. So we're interested in putting as many possible samples onto a chip. They're expensive and difficult to set up, so we want to put as many samples on as possible. And we want to balance the number of samples we put onto a chip with the coverage that we get to give and the multiplex scales, because the more SNPs we put into the multiplex, the more sequences are being occupied on the chip. And also have this sort of quite good cartoon in there, sort of YouTube animation, which demonstrates the need for having plenty of coverage. But we're dependent on the number of pits there are in the chips and how well they're occupied. So are there coverage outliers? And can we set a minimum coverage limit? And one of the problems we've had in setting up the validation program that I'm going to go on to explain is that the amount of samples you can load onto a chip and the amount of coverage you get is a sort of moving landscape. So this is increasing as the chip manufacturing process is better QA'd and better optimized. One other consideration that is worth mentioning is that SNPs are binary. They're not like STRs, of course. And Ion PGM is a very sensitive platform, so we do need to consider mixtures. We're going to see mixtures, and we're going to be compromised in our ability to detect and analyze mixtures because we're dealing with binary polymorphisms. So allelic balance becomes important in this context. So we need to say what are the patterns of allelic balance in the SNP set, and does this impinge on our ability to detect mixtures? So the three studies that I'll describe are the work we did with Victoria Police Lab, which combined 5 snapshot PCRs, nice simple system. We just do the PCR as we've always done for snapshot, and we combine them all together into a single library preparation step. And we didn't balance them in any way, we just put them together and used elution columns to purify the products. And a nice simple program of 3 samples, 3 input amounts to test, and checking concordance with previous snapshot results and Sanger sequencing when necessary. The third validation program was a chance to look at a prototype version of the LT identity panel, which we had access to. And we decided to use a very simple NIST-based validation setup framework, which I'll describe in a minute. And the problem that we had was we had 4 different chip types available to us, but we kept it simple in terms of the number of samples. And an important step following the Vic Police study was to use concordance checks with public data from 1,000 genomes and complete genomics. And the difference, another important difference that should be highlighted between the 2 studies here is that Ampliseq technology was not available to the study made in Australia, and we were looking at Ampliseq designed primers in the second program. And lastly, we've used the same framework for the middle study to look at some ancestry markers that we've developed from the ground up. We've developed from scratch. So very much like Sally Ann's program of looking at the SNPs that they want to look at or the markers they want to look at, we've done the same thing. So moving to the Vic Police study to start with. The SNPs that the Melbourne lab was interested in looking at were SNPs that provide ancestry inference, identification, and the first step into EVC prediction that's appropriate for the Australian demographic. So eye colour prediction is going to be important for a mainly European, predominantly European population. But equally, it's important to distinguish between the four major parts of the Australian demographic of European, South Asian, native Australian, and East Asian. So we combined a prototype ancestry set called Pacificplex that distinguishes Melanesians from East Asians quite successfully, the 34-plex ancestry marker set that's already well established, Eurasiaplex, which is designed to distinguish South Asians from Europeans, and then we had the 52-plex for identification, and Irisplex using Manfred Kaiser's private designs. So as I said, we just amplified 3 controlled DNAs in the 5 PCRs, combined them after a quite quick column elution purification step, but no quantitation, and pulled these for end labeling and putting into the library preparation. And we've got a variety of different sequence. These are sequence levels, coverage levels. We've got a wide range of different coverage levels. And you can see here that Eurasiaplex dominates the top 10%. So this is a multiplex that needed reoptimization in Australia and wasn't successfully completed. So it dominates a large part of the coverage output. But having said that, the coverage was very high. We've got a very large amount of coverage from these PCRs and running these 3 samples on the chip, 314 chip. In fact, we obtained 3 times more coverage than we expected from this chip. So this augured very well for the coverage we can expect. And the other important thing is that the template amounts didn't influence the coverage that was obtained. You might argue that these template amounts are quite similar from each other, but the fact was there was no link between input DNA and coverage. Allelic balance was okay, but not ideal. There's quite a few heterozygotes that have allelic balance in between an ideal 50-50 and 100-0, you could say. So there was a few outliers that were between 10 and 30% ratios. This is comparing the reference allele to the alternative allele, 70-90. And these are difficult to interpret, of course, because you don't know if you've got a homozygote with some spurious alleles called, or you've got an imbalanced heterozygote. They're very, very ambiguous in terms of calling the genotypes in a reliable way. But the outlier allele frequency ranges did correlate with low coverage. So you see on the bottom charts that in the middle range between the midline of the chart, of the 3 charts, and the left and right-hand side, most of the outlier SNPs have very, very low coverage and sitting in the middle of the range. So this very small proportion of SNPs could be, that are consistently imbalanced, could be discounted from any analysis you do. You just take that data away from any interpretation you might make. Routine forensic scientists might not want to go to court with just some of the data rather than all the data, but it's something you could do. Genotyping concordance was quite good, but it did highlight some problems we have with snapshot. So we reverted to Sanger sequencing as a sort of third strike, if you like, on the genotype. But as I said, the overall concordance was reasonably high, 98.5. And you know, there are well-established polymeric tracks that cause misalignment that corresponded to one miscall in the ion torrent, the second row here. And we did have one SNP that doesn't have any apparent context sequence problems that gave ambiguous genotypes between all 3 systems. This can sometimes happen without an explanation in this case. So the conclusions are that PCR multiplexes, as they are, can be used unmodified and provide you with quite good quality data, really. You know, this is something you can do if you're interested in developing new markers or you want to test the SNPs that you already know and trust. There were 4 or 5 discordant genotypes that had low coverage. So this really allowed us to suggest that 20 times could be a suitable minimum because 4 of them had coverage of 13 times or less. So that suggests a value that might work. Although the average coverage was very high, there was some variability, as we've seen from those charts. There's a variation between coverage with any one multiplex. And we put this down to some extent to the absence of Ampliseq. We think Ampliseq balances the coverage very, 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 very well. And of course, snapshot could be better balanced. So we can, this is work in progress, but it works perfectly okay in a CE context. So, we could balance it better. And we're not certain at this stage whether applied biosystems balance the PCRs in an equimolar way or whether they adjust the molarity. This is an open question, I suppose. It sort of invites an answer. I don't know whether we get one. Yeah. And as I said, imbalanced heterozygotes did correlate with low coverage, but not all low coverage SNPs were imbalanced. So, really allele read frequency thresholds, where we set limits for the balance between each of the alleles in heterozygotes, might be our preferred approach. That threshold might need adjusting for the worst SNPs. Moving on to the validation where we looked at a pre-commercial kit from LT. The kits have changed slightly from prototype to the finished signed-off version. Neil's already explained some of the numbers and the way they combine. These are the final products that we can buy from Applied Biosystems now. So 124 identification SNPs and 165 ancestry informative SNPs. And we were working with a prototype that had more. So I just highlight that fact that this has been changed. So 45 autosomal SNPs have been removed and 9 Y SNPs have been replaced. And just this year, the three last Y SNPs were swapped out to give a better, higher clade sort of combination, if you like. But we think that these primer sets are based on previous snapshot designs, but also they buy into the Genplex work that Applied Biosystems did some 8 years ago now. So primers were designed for the Genplex for the 49 over 52 SNP for ID SNPs, and as well as HIDS 45. And we did have a successful collaborative exercise that Copenhagen organized looking at Genplex. So these are robust primer designs. As I said, we wanted to try and adopt a very simple validation program. It's complicated a simple validation framework to test it. So NIST recommend a very simple microtiter plate format, if you like, of dilutions, simple mixtures in a series of just 5 different ratios, some NIST control samples, if they're available. Of course, these have been typed for, in this case, STRs already, and to put in some non-probative challenging or degraded DNA samples. 2391B has now been superseded by C, so we opted to replace these with the standard controls that are available in all labs. And we also added 7 genomic controls that have already been typed by 1,000 genomes in complete genomics. So that's 2 independent large-scale genomics projects that have their data online, and you can cross-check your genotypes from your system with what's available with their system. So sensitivity was tested with simple dilution series of 2 samples, and we had access to a 12th century or a 5th or 6th century skeleton from some samples that Walter described this morning. Concordance was interlab and intralab, so between and within laboratories. We did concordance checks against the Coriol data. Mixtures were very simple male-female staff combination in 5 ratios. And the qualifying run, which strictly speaking should be done within a laboratory with different operators, was done between labs. So we really tried to make sure the protocols were as closely matched as possible. This is just technical stuff, so we use variant caller, somatic and both somatic and germline parameters. And we were interested in 4 key parameters. Coverage, of course. Coverage is everything. Allele read frequency, which we've already seen in the work we did in Australia, so the balance, if you like, within heterozygotes or between the alternative and the reference allele. Base misincorporation, so how reliable is the sequencing reaction. And strand bias, which Bruce mentioned this morning. Do we see a disproportionate number of sequences from one strand direction compared to the other direction? Coverage. So this is all the SNPs in the 169 panel we looked at, ordered in terms of their mean coverage. And the ones with the black dots are Y markers, so with half the template DNA, they have lower coverage. That's not a surprise. But there's also some autosomal markers, autosomal SNPs in amongst those as well. But generally speaking, the coverage was very, very good. These are mean values, so they include low-level DNA as well as optimum DNA. But this represents a much more even gradient, if you like, between the lowest coverage and the highest coverage. I think Manfred's right. I think big slides take a long time to progress. So there's a big graphic coming up. So maybe the computer's a bit slow. Thanks. So when we move from mean coverage to coverage per run and coverage per SNP, we get this rather complicated sort of heat map, but it's been ordered in terms of the lowest coverage per run, or per analysis, I should say, and the highest coverage per analysis going from top to bottom. And from left to right, we've got the best, the worst SNP in terms of coverage and the best SNP on the right-hand side. And the Y SNPs, slightly less analyses because we had less males than total samples, of course. But the important point to mention here is that the top of this these charts it's stalled again. Includes mainly samples that are amplified with less than 100 picograms of DNA, degraded DNA, and also runs where we really put more samples on a chip than would be ideal, would be optimum, because we had this problem of knowing how many samples to load. But it still suggests this 20 times minimum coverage should be sufficient for the vast majority, something like 95% of the SNPs in the panel. Allele read frequency balance was pretty good. These cluster around the midline of 50-50 for the heterozygotes, with some obvious outliers. So how do these equate with previous studies? Well, as I mentioned, Klaus Borsting and Niels Morling had done a similar kind of validation of the same panel. And the ones in grey are the ones that were identified by that study only, but not by us. The ones in blue were identified by both the Copenhagen study and our own study, and there are two other SNPs that were identified as outliers for allele read frequency by ourselves only. So you would expect that you would identify the same bad SNPs and good SNPs in terms of balance, but this is not the case. So it suggests that we may all have to do our own validation program to identify what happens with heterozygotes, which is a bit of a daunting prospect. We can't validate for everybody's sort of data, if you like. This is the paper that represents the study from Copenhagen. Base misincorporation was very, very good. Very, very low rate of misincorporation. Generally speaking, it was about no more than about 0.25% misincorporation, so the wrong base where it should be, with some outliers at the right-hand side of the chart, but no more than a maximum of 3% in 1 or 2 cases. These are Y SNP reads in female DNA. So I don't really think this is drop-in, but it's the closest we could get to drop-in. And it's really only a total of 34 wrong bases, they shouldn't be there, obviously, in females, amongst 2 million sequences. So this represents a really, really low level of infidelity, or low level of, infidelity is the wrong word to use, isn't it? Really low level of incorrect base calls. Strand bias, well, we set thresholds of 0.25 and 0.75. So, in other words, nothing exceeding a quarter of reads in one direction and three quarters of reads in the other, that ratio. We did have quite strong outliers, so it's these 9 SNPs listed here. When we look at IGV, which is a sort of open source sequence reader, we saw some quite unusual strand bias patterns in 1 or 2 SNPs, which we have no explanation for. So in this case, there's almost no reads in one direction at all. We don't know why this is. It could be something blocking the alignment in one direction, but we just put this up as an observation. Now, this is okay, but we found that it affects the calls. So this is an example of a SNP where we've got less strand bias, but the reads in one direction are marking indels where there should be SNP alleles, and again, we have no explanation for this, but it really highlights the fact that strand bias is an important thing to measure. Concordance. Well, we really only saw one discordant result, and it was in a single sample, and it also has well-recorded misalignment problems because of polymeric base. The 2 Y SNPs at the top of the table are incorrect bases. They'd be identified as a missed call anyway because they shouldn't be there. This is an extraneous 3rd or 4th base. So we saw really, really good concordance values. Very, very good indeed. This is this example of polybase again, causing misalignment and causing a missed call in that 3rd row. Mixtures. Well, mixtures were good until we got to the 9:1 ratio. We don't quite know why this one is not quite so good as the others, but there were clear displacements in terms of allele read frequency. So this means that although we're dealing with binary markers, so we have compromised ability to just differentiate 2 people combined at random, we still see unusual patterns at these ratios. So they should make themselves known that they're mixtures, and they should be obvious and easy to detect. Sensitivity. Well, we did limited experiments with sensitivity, but it's true to say that really for 25 picograms and 50 picograms, we really only got at most 10 to 12 SNPs dropping out. So for the optimum input concordance studies, no more than 1 or 3 SNPs gave no calls or dropped out. By dropout, I mean they had a Q value below a certain threshold. And all these were outlier SNPs, so there were SNPs that gave strand bias or allele read frequency displacements, etc. But in low-level samples, as I say, no more than 8 or 12. So this really is very, very good. We did also look at what kind of output we got. These are untrimmed sequence read distributions in terms of lengths, and you can see the obvious thing that low-level DNA does provide less output from the library preparation, and from the PCR, I should say, and you do get a primer-dimer peak. An obvious primer-dimer peak at 50 base pairs. So this means that the multiplex is less efficient, which is sort of what we would expect. Just a single ancient DNA sample, so it's a limited experiment. We managed to get 450 picograms from this, or not necessarily this skeleton, but one of the skeletons from this site. And we did do the 25 PCR cycles plus 25 PCR cycles and five library reamplifications. And the condition of these skeletons is really quite good, and it's written up in Walter's paper that he showed this morning. So this is not at all possible to read, but this is the the results in terms of SNPs listed by coverage. So the best coverage SNPs are at the top, and the worst coverage SNPs are at the bottom. We've got a lot of no calls and a lot of quality values below 0. So these are no calls at the bottom. But there was quite a large number of SNPs that gave coverage above 100 times. So this is a really robust technology in terms of coverage from a challenging DNA sample. And when we do random match probabilities, we get 5 orders of magnitude better values from these SNPs than we would do from the best STR set. So again, the quality of data we're getting is very, very good, even if we just take the top SNPs. There's more good news when we align these in terms of amplicon length. They do kind of tally. So I've marked the average for every 20 SNPs. So it progresses from 90 down to 137 at the bottom. So there is some kind of correlation. But as I said, the good news is that LT have really worked hard to reduce the size of the amplicons in the current version. So when we put these in order, this doesn't correlate to the chart on the left-hand side. We really see that they've been working very hard to redesign the primers to bring down the amplicon lengths for at least a third of the markers by an average of 57 base pairs. So that's really good. That means that we can expect better performance with very degraded DNA in the future. So to summarize what we're getting with good and bad SNPs, we have 5 that we think should be removed from the panel because of discordances or misalignment problems. And the 2 that are underlined are still in the signed-off panel. So they, at best, need to be treated with caution or discounted from the analysis. Then we have various others that have outlying characteristics that may interfere with data interpretation, if you're looking at mixtures or degraded DNA. And then those that have some outlying characteristics but don't interfere with reliable performance. So the conclusions are not all SNPs performed equally well. That's no surprise, I guess. 5 gave genotyping discordances, both interlab and interrun, and 2 are still in place. Several other SNPs have outlier characteristics, so things like imbalanced allele read frequencies, those that were identified by Borsting, the 2 that were not identified by Borsting but identified by us. So really, we will need to validate this allele read frequency characteristics ourselves in each of our labs. Concordance rate was the best we've seen, really, really good values. From the limited experiments with low-level DNA, we've got very high sensitivity so far. So experiments we do in the future, I'm sure, are going to be very, very good in terms of the quality of data we get. Nearly half the SNPs gave coverage over 100 times, so well in excess of the minimum coverage for reliability. And 57 of the SNPs in the current identification panel have been shortened further. Harmonizing chip loading has been our biggest nightmare, so it's difficult to do, and we have to sort of tread carefully as we introduce this technology into routine labs, and there's no easy path, I'm afraid. But it did cause a lot of variability in this program. And the idea of identifying the allele read frequency outliers is really that they could be discounted when we're analyzing mixtures. So you've got what looks like a mixture pattern, but you actually, from those that are already fairly imbalanced. And I make this point that the genotyper that is used for the forensic kits uses somatic parameter settings, but we found that germline parameter settings are better. The final message is that Euroforgen is working on a mixture analysis system for Ion PGM data, so we're hoping to introduce likelihood-based systems that will allow us to sort of make a formal analysis of mixture patterns. Okay, so just 2 or 3 slides to finish with on the Ancestry Panel work, which is still ongoing, so it's sort of early days. The idea of this panel is to provide some balance between the 5 major population groups worldwide, but also with an emphasis on being able to reliably detect admixture. By admixture, I mean people with co-ancestry from mixed parentage. as this is a sort of dominant feature of most urban demographics. The assay conversion rate was very, very good. So LT provided us with 125 of 128 SNPs, so that's really good. So far, the concordance rates are very, very good, very high. The interrun rate is 99.98, so that's very high, just one SNP giving problems. Interlab, there are 6 SNPs that are giving problems, but the reason for the discordances have been identified in each case. And concordance with reference data from 1,000 genomes and complete genomics varies a little bit between the two because they don't always give reliable data. Complete genomics has revised some of their SNP data, but pretty good, above 99%, which is the important thing. The 6 discordant SNPs in 13 genotypes were mainly homopolymeric traits, so perhaps the strategy in the future is to look very carefully at the context sequence and handle with caution or handle with care these kinds of SNPs. And we're working with LT to try and have some form of retrogressive analysis so we can go back to IGV-type data to sort of identify these as problem SNPs. 8 no calls, 4 low coverage, 1 population-specific flanking indel, 3 didn't pass quality. And the final message is that we do see some low coverage that's confined to one population. So all the samples from one population have below average coverage. And this could be something like untracked primer site SNPs. So we need to think long-term about redundant primers, perhaps, for particular populations. OK, that's it. Thank you. Just going to finish with a photograph that one of our lab people took yesterday. Yeah, this is the anatomy professor who's obviously been working too hard. OK, so acknowledgements to everybody from Santiago, to Mayra and Walter from Innsbruck, Teresa and Peter from Cologne, Matt Phipps from LT, and David Ballard from King's College London, who helped us with the data analysis in Santiago. That's it. Thank you.